So we've seen many, many great references. It was great all day yesterday. Today I'm writing down references, and then I realized, oh yeah, they're in the proceedings. But point is that there's so many great things that we've learned together so far that kind of pulls this all together. I think we'll probably say in our summary statements, led by Natalia later when the chairs all come up here, one of the things that we're going to have is a call for action that we need more data on the pet food side of things, the nutritional immunology side of things, because we just don't have it. But what I wanted to mention to you today in my talk, just as a kickoff, is I actually did become very interested in this space, a lot because of Ebenezer. And many of you heard his great talk on immunonutrition yesterday. I actually read this paper when you published it, Ebenezer. So this one's 14 years old now, 13 years old. But it was like, oh, that's what it's all about, as an internist that cared about all these things but didn't know much about it. So actually feeding instead of just pushing pills, which some internists do, was a kind of a new beginning for me. And so if you remember from his proceedings and his talk, I'm like, well, how am I going to build on Ebenezer in my talk? And then I realized, well, he wasn't going to say a whole lot about systemic diseases, and that's, of course, what I'm most interested in as a PhD parasitologist, infectious disease person. So what I'm going to do now for the next 28 minutes or so is really just show some of the work that we've done, work that others have done, obviously, and some of that call for action in the future. Because if we can move the needle systemically, obviously keeping the bugs out of the body would be the first step with our leaky skin and leaky bowels. But once it gets through, if we had better systemic immune responses to lessen disease and or mortality in some situations. We got a big boost, right, with the COVID-19 thing. Yeah, SARS-CoV-2, immunonutrition became very interesting to many people. So that's what I'm going to do with you guys today. I don't work for Purina or the Purina Institute, but just those of you that are visiting that didn't know about the new version of the clinical nutrition book, it is not a branded or product-based book. It's actually funded by them, and we thank them for the funding. But all of us that participated in chapters, which many of you are in this, we didn't mention a product one. And so, again, I don't mind sharing the QR code because all of my residents and all of my students are now using this as part of their nutrition training. But the other reason I brought it up, because it is a lead into my talk, I have actually spent most of my nutrition space in the supplement world. And so I became quite interested when I first met Christoph and some other nice people from Nestle. I'll share a story a little bit later, that perhaps it was worthy of studying if you're an infectious disease person. But it was kind of fascinating, right? Because those of you that have been doing probiotics and other supplements for a long time, it's been a bit of a leap of faith, right? That they actually do anything. And we know it's a multi-billion-dollar industry in humans with very little data to support most of what people are doing. And so they're just spending all this money in the health food stores, right? And so I got quite interested. So thank you for letting me do this, Purina Institute. But, you know, it really starts with making sure that you're actually buying, quality products, and it's a really big part of it. And so when we're thinking about the immunonutrition data, we saw some great individual papers. I learned a lot today. I thought of some new studies that I want to do based on what I've already learned. It's just amazing when you try to do a meta-analysis in veterinary medicine on immunonutrition. It's a little tough, isn't it? We have some pretty good review articles. I'll talk about GI review articles probably before we're done today. But to have a true meta-analysis where you could look at 12 studies on the same whatever and actually develop any conclusions is actually quite lacking. We don't need definitions for this group, but I've spent most of my time in the probiotic space working with a number of different bacterial organisms. So we need to do this study again. Those of you that are young in training that want to actually go buy a bunch of products and culture prove what's in those. But Scott, when he did the two, Dr. Weiss did the two studies kind of back to back in 2011, 2014. It really emphasized, at least in the United States, that just showing that your product is safe is pretty much all you have to do, right? You don't have to actually show it's effective. And you don't even have to show that it has its published or suggested number of live organisms at the end of the shelf life. And so I am glad to have the 4,000 people that are on the line as well. Just to remind people, before you buy something, make sure that you ask that company, can you show me your organism numbers at the end of the shelf life? Not just at the beginning, right? And I don't know if any of you are in this room, but have had your probiotic study fail because you didn't have any probiotic in your product at the end of your study. Nobody wants to raise their hand? Yeah, I've lucked out so far. All the ones that I've done, we actually were able to grow live organisms at the end. But it is a bit of a buyer and researcher beware field. And so that's my first step if I'm going to do a clinical trial with somebody, is I'd like to see your stability stuff. And so that's a first question. And then, of course, I think if you read my how to use supplements, I won't use a supplement that they haven't published something in the peer-reviewed literature. And so that's how those of us that are the gateway to a veterinary school pharmacy. That's minimum standard. I'll talk to any drug company, any pet food company. I want to hear what they have, right? You want to hear the new stuff. But if they haven't published a word on their product in a peer-reviewed fashion, it's not making it to Colorado State University. So that's my two levels, right? Safety, stability, published efficacy. Otherwise, I'm not interested personally. But as you guys know, we've been talking about metronidazole a little bit already today, but wouldn't it be great to do a study that at the end you didn't have any live organisms and still try to figure out how to publish it? Yeah, we need to make sure those companies give you good stability data. You know, Al Juergens at Iowa State, he's not here with us this weekend, but Al's been really active. Obviously, we all interact with Jan, as we mentioned, and it's just been great to see us gather more real science. And it's really you guys more than me, because I'm just an infectious disease guy, but it's really been fun to see now we're showing local effects. We can change a leaky gut. We can measure a leaky gut, things that we weren't doing 15 years ago, being midway through my career. We weren't doing that stuff, right? And so it's just been so great to see it over the course of these days together. It's just fantastic. But what we haven't had quite as much, we're getting some good clinical trials. It is still kind of funny, though, that you can pretty much sum them all up in 2 review articles when it comes to probiotics in the bowel. It's really kind of sad, but easy because you don't have to read that many papers. It's good, though, to see that we would continue to move that forward. Obviously, we've talked today also about allergy. Skin disease is great. In this particular meeting, since we're doing GI stuff, we won't talk about the gut-brain barrier, but those things are great as well nutritionally. Things with medium-chain triglycerides, probiotics, bifidobacter group that might, change anxiety and or stress in animals. It's just really great to see that these living organisms, probably tell I'm going to talk only about probiotics and symbiotics from here. It's just great to actually have some products, because it is ultimately we need to actually use these things. And so as a scientist, I love to see the laboratory-based things, but it's also great to see it publishing with products that we can actually use. But what I'm going to focus on now is just the systemic stuff that we've been doing and a number of the others to try to get a couple of, I think, key points across before we bring Allison up to the stage. So I'm going to tell you the story about one of the organisms in a couple of minutes, but what happened for me is I wasn't using probiotics much, even though I was an internal medicine person that's interested in gastrointestinal disease. I just hadn't been that convinced. Rarely 20 years ago would I have sent a client to a health food store to buy lactobacillus. I just hadn't drank the Kool-Aid at the time. And so that's when I met Ebenezer and some of his buddies, Christoph and Jaleel, 2 researchers from the Nestle side of things. And they actually told me that they had an organism that might stimulate systemic immunity. And that kind of perked up my ears. And so honestly then, this is from a long time ago actually, I was just kind of thinking about, wow, if there was really a systemically immune-stimulating probiotic or immune-modulating probiotic, either up or down, but with infectious diseases, I was thinking stimulate in a good way to control infections. It's like the list got pretty long, right? And so you can think of all these things that, man, if I could make them better faster if they were sick, or preferably if they were healthy, if there was enough data that said that feeding something would make them less likely to die of COVID-19 or whatever, then I might actually prescribe it. But that's what's been fun being in the blend between a clinician and a pseudo-researcher, is really my end game is always wanting to know what can I use to keep animals healthier and or treat them, because I still think myself as a veterinarian first. But as you guys go through your careers, you'll probably do it the same way, since many of you guys are practicing veterinarians. So I actually thought this was a really fun paper to read if you guys haven't read this one. It really did stimulate COVID-19, obviously, SARS-CoV-2, stimulated a lot of interest in all sorts of things, but probiotics were one of them. And actually, it's kind of interesting. There's a lot more data for human studies than there are for veterinary studies at this stage. But it was really kind of fun, because then it makes you start thinking, right? Well, parvovirus now is going to tie into chronic enteropathies. Thank you, Stefan and others. Giardia, all these things. If we can make them less likely to damage that epithelial barrier, then we're probably going to do okay. Just like in animals, getting a probiotic effect fast enough to actually treat a human, it's kind of been one of the holy grails, right? Because we don't all do preventative things. And so then now you're already sick. And so actually finding something that works fast enough, and that's maybe where our work with postbiotics, will be the next holy grail, right? Is we don't have to have living organisms. If we can actually just use those byproducts to help us, that'll be great. But I just wanted to use this review article just to remind you that there is actually a pretty good body of work saying that probiotics might be helpful in systemic infections like SARS-CoV-2. Yeah, I wanted to just bring this one up real quickly. The Lactococcus lactis organism I didn't know much about until reading this particular review article, but it's pretty interesting in the things that we're all caring about. Once you've gotten into the body, can we do something about it? So our barriers failed us. Now we got to try to get them out of the hospital faster. So here's an interesting one. I know that Ebenezer mentioned it. Dr. Gore was a nice Nestle researcher. We got to work together a couple times. This was great. The colostrum, I buy it, I believe in it. Then, obviously, they did the same thing with dogs. You saw both of these references yesterday. So kind of starting to believe in immunonutrition. But if you're thinking about they're sick today, well, that's not going to help you today, right? It takes time for these things to work. And so I kind of started drinking the Kool-Aid with them a little bit, starting to read papers on other bacteria that are known to have systemic effects. This one, Anthantis, is a really interesting organism. And just to kind of tie it into our lectures this week, is they did look at an enteropathy type disease, but then they looked at a skin disease, and then also an undescribed inflammatory disease with chronic fatigue. And so we're just getting more evidence that says it's worth you guys that are early in your career continue to write grants to try to prove this stuff actually works. It's not just our sponsors. There are other great companies around the world, and that's what's fun to do these non-denominational meetings. It is great to see people doing work to try to show systemic effects. Obviously, Slab 51 has probably had about the same amount of research, maybe a little bit less than SF68, one of the other organisms that we look at a lot. But it's just fantastic to know that we have some of these effects. They just need to be better characterized. So this is when I met Jaleel and Christoph, and a couple of you know the story already because I've been lecturing for a couple of years, but it was kind of interesting. They came up to me at BSAVA, I think in Birmingham that year. Kenny and I were probably doing something the night before, but they came up after a cat lecture and said, oh, you have some of the early immunological tools for measuring cat immune responses. They say we have a product, a bacterium that will be a product, so it hadn't come out yet, but it's Enterococcus faecium strain SF68, which you might know was discovered in 1968, so it has a lot of data behind it. And they just said, would you read a preprint of a puppy paper? And I did. They gave it to me. It wasn't a PDF. I don't think Adobe had been invented yet. But I took the little pieces of paper and I read it on the way back to Colorado. And I was quite intrigued by it. And that work, and they showed, a pretty strong systemic B cell stimulation. Not a lot in the T cell side of things. If you've read this one, it's a little bit ancient now. But it was very interesting, very similar to some of the things showed with SLAB 51, as that we can move the needle. And so I read it, and I thought, well, this is pretty cool. I'd be happy to do the cat stuff with you guys. I'll show you a few of those studies here in a couple of minutes. But as I was thinking about it, either the colostrum papers or if you look on the axes, these are weeks of supplementation before you see separation. I was kind of back to well, that's kind of cool, but I know I don't need to have an immune modulating supplement to vaccinate. That's why we put that extra log of vaccine, right, of virus in our vaccines. The vaccines work fine. So it's not like, I have to supplement puppies with colostrum or a probiotic to vaccinate effectively. They're immune studies, right, just to show you can move the needle immunologically with these supplements, and so that's kind of cool. But then I'm like, well, now I'm getting bored again because I'm a clinician, and I want it now. They're sick today. Even the chronic diseases, Domenico with his life with chronic skin infections. But that still takes time, right? And so if it's really not doing it until 26 weeks, that's just not going to do it for me. Now, maybe as a prevention, if you have a disease you know that they get quite frequently, that would be beneficial. Plus the fact that they showed mainly B cell responses in this one. Tools have changed a little bit since then for the T cell side of things. And so we didn't really know, right? And so I was kind of interested because it seemed like fun science projects for my residents and students, but wasn't really that turned on. I don't think many of you have seen this work. I showed it at DACVIM a few years ago, and then we went into a longer clinical trial, and it will be combined in a paper that will be hopefully accepted. We'll see what you guys think under review. But we had some leftover beagles. And I do like to mention that Colorado State, Alice and I, since we work a lot together, I want to let you know that our group, my Center for Companion Animal Studies, and Dr. Dow's group, the Center for Immunotherapy, we don't euthanize animals. And so when we do animal studies, we do adopt them all. And in fact, I would like to invite some of you, if you would like to come by Colorado, there's about a week of skiing left, and while you're there, I will give you a beagle. And a cat. In fact, if you want 2 cats, I'll give you 2. No, we do a little purpose-bred animal work, but we do adopt them all. That's kind of where we've settled out to what I could personally tolerate as an investigator. But we do adopt them, and they all do great. I can't wait to show you one of our studies in a minute. But it was just one of those Saturday nights you're bored, so you're thinking about, what study could I do next? You guys do that, right, on Saturday nights? Yeah, and I had some leftover beagles, and I was still thinking about the puppy work, and it's like, well, those are growing puppies, right? They're not immunologically mature yet, and so I thought maybe there would be an opportunity to just do it again in some young, healthy adults. And so I didn't have that many. I just had 7 left over that day. But I'm like, well, they're getting ready to be adopted. Why don't I write a protocol? It happened to get funded. iCook has no problems feeding probiotics to healthy beagles. So it was an easy one to approve. But just to be clear, we're using their own baselines as their control. There is no control group in this one other than their pre-supplementation. So we did want to know, does it really take 26 weeks to separate, right, between supplemented animals? These guys, we justified the lack of a control group by the fact that they were young, immunologically mature at that point. Other studies I'll show you in a couple minutes. We use control groups in our studies as well. But this one I chose to compare to their baseline. And so we've already talked about a few studies in the ancient days. Remember Con A? Yeah, they were even teaching concavenavalin A when I was a vet student, and that's before there were computers, actually. But it was kind of interesting. In this study, we showed pretty clearly at 4 weeks, really pretty significant increased responses to mitogen, this mitogen compared to their pre-samples. And so I felt that that was fairly intriguing. I think the work, and we have done this multiple times since then, this is just that published abstract that I said to be slide-worthy, you had to have an abstract. We've actually shown that B cell side of things is pretty pronounced, and that could be good or bad, obviously, depending on the disease you're working with, but overall, if you can move the needle on total IgA, maybe B cell expressing lymphocytes in general from many bugs, that would be a good thing. And at as early as 4 weeks, statistical differences. And then we also saw some changes with T cytotoxic cells. And so we got a little bit now more intrigued because it wasn't 26 weeks, 10 to 26 weeks. Now it's somewhere between 2 and 4 weeks. I've already admitted this multiple times, but I was a little embarrassed with myself. I just don't know why I didn't bleed the dogs on week 2. It would have been logical to do week 2 as well, but I didn't. And so honestly, when people ask me, how long does it take? Somewhere between 0 and 4 weeks is what the honest answer is, because we have not repeated the experiments at 2 weeks. Maybe we will if they have a good research budget year. The point is we have not with SF68. But I think you'll enjoy the paper when you see it. There are a lot of other numerical differences between 7 dogs, and it was enough that made me feel that we should do a clinical something. You know, I love normal beagles and normal cats because we can adopt them and they're kind of controlled, right? You can feed them the same diet and makes it a little less variable than in the field, but it's not as realistic as the field because we all do different things. You just need bigger sample sets, right, for the field studies. But I wanted to share with you, I was trying to rack in my brain, I was talking to the derm people. Thank you, Domenico. Thank you, Galia, wherever you are. There you are. Yeah, so hanging with the derm people because they're much cooler than internists. And I learned from them that there are a lot of things that they could use an immune-stimulating probiotic. And so after many, many discussions, which actually did involve Emily Cross as well, we actually well, why don't we pick the generalized Demodex dogs? And I don't think any of you have seen this data before. It's the other half of this paper that's coming out. So I'm not going to show you a lot of data because I want to jeopardize publication. But this one was, okay, well, what do we have? An unlimited supply in Colorado, and that's the pit bull terrier. We did not genetically try to characterize them. They just had to look like a pit bull terrier. And we didn't want localized Demodex because it can change, so readily. Put oregano in their left ear and localized goes away. So bottom line is we wanted generalized Demodex. Worked with the dermatologist because I'm obviously the internist on this one. And so we had plenty of dogs. They all needed to be rehomed. My favorite one of the study is the little pink pig. Yeah, that's Tulip. I'll show you some of her after pictures here in a little bit. But you know what? The hardest thing on this particular study was convincing the derm resident that it isn't a Demodex study because I wrote it for using Advantage Multi. Didn't that make your toes cringe? Yeah, I know. What? Why did you use an inferior drug? Well, that's the same thing I had to talk to the derm resident about. I didn't know how quickly it kicked in between 0 and 4 weeks, and so if I gave something like Bravecto or other products in that space, they'd get better too fast. And so remember, this is an immunology experiment. So we did use Advantage Multi weekly. It was great that it was a study because that way I could give all of them Convenia and not feel guilty. Jan's not listening, thank God. But if you give an antibiotic, people throw rotten tomatoes at you. But again, we're trying to standardize it, right? So they did all get Convenia shots. They all got Advantage Multi weekly for 12 weeks. And then what we did is we did randomize. The derm people didn't know which dogs were on the probiotic, and the people doing the skin scrapings and the scoring and all that were blinded. So this one was placebo-controlled. We ended up with just 5 in the placebo group and 8 in the probiotic group is where the data fell out by the time we showed it as an abstract. But if you hadn't heard me talk about it before, supplementing with this probiotic that we had shown some effect on both B and T cells in the previous beagle work, we did have some evidence at the 0.1 level, so it didn't make 0.05, but in that 90% confidence intervals that we had improved mite kills and speed to clinical resolution. So I think we're going to submit it as is, just because that budget has been spent out now, so we won't be adding any new dogs to this study, but those of you that are looking at continued Demodex study, Galia or Domenico or your students, we have some at least pilot data that suggests it might be a decent model, just because, at least in the States, we have lots of generalized Demodex pit bulls. So we've concluded in that paper that you haven't seen yet that it is evidence that there's a clinical effect of what we saw in beagles that were healthy and of an age that they probably had a normal immune system. So that's our dog stuff. But actually, remember back to the story when I'm talking to Jaleel and Christoph, it was actually about doing cats. And so this kind of came on later. So we started doing the cat stuff about 15 years ago. And that's when I really got to work a lot with Ebenezer. But before I close off on that one, the other fun thing, those of you that work academically, is that it was really cool to also use this work to have students understand socialization of animals, appropriate socialization of animals. And so we're working on a veterinary medical education paper from these dogs as well. And Alina Contreras, who was my shelter resident at the time, a PhD student, she's at Long Island now, or just left Long Island. Bottom line is Alina did this work, and it was amazing. She would have picnics for these pit bulls. Colorado, we're a little weird. Yeah. Anyway, so we have pit bulls for pit bull picnics on Saturdays. You can come by when you come visit. So now I know these really smart people at Purina and Nestle, and I'm like, okay, we'll do the cat work. Julia Veer, who many of you guys know, did her PhD at CSU, 2 generations before Quimby, I think, maybe 3 generations. Bottom line, she's a nice young researcher, and she worked with the Purina folks. This is an older paper that you can pull later if you want to read it. Didn't show a lot in this paper other than percentages of CD4 positive cells went up over time. And so then as we're chatting about, well, what would you do if you had a probiotic that might change T helper cell percentages, it kind of came back to maybe a viral disease. And so that's where the work came from, where we looked at herpes virus first. We have new work looking at calicia as well. I do have a sanctuary that we're working with that has over 800 cats, and many of them have stomatitis. So that was a nice opportunity to study now in the field. But what we've published so far, first and foremost, this is back with SF68 again. We've done most of our work with that single-strain probiotic. We actually showed pretty convincingly, and the microbiome for this one was done by the Purina researchers. Pretty much everything I'll show you after this will be Jan did the work. But bottom line is that we did show in this stress model, and we call it a stress model. I can't remember now who was talking about cat stress other than Jody. But the point is all we do is change their housing. Yeah, they're in the same gang room, and then we change them from cages into the gang room, back into cages, and showed that if they were supplemented with SF68, they actually had better diversity. So whether or not that's an immune-enhancing effect or that's just the probiotic effect is probably the probiotic effect. But what was the interesting clinical part, those of you that haven't seen this one in a while, is those kitties that were randomized into the SF68 group had much less reactivation of herpes signs. If you're going to do any of this type of work, I'll give you a couple of hints. If you want to study respiratory, do your challenge model, your inoculation with intranasal. If you want to do eye studies, do your inoculation in the eyes, because it really is different depending on how you inoculate them. And this is all CSU work, not Purina work. They obviously wouldn't give cats herpes. That was me. But bottom line, they were vaccine cats, and so they were left over from vaccine studies. And then they got readopted. But why I wanted to give you the lead in, and I'm only going to go about 5 or 6 more minutes, Allison, if you want to get ready. No, I'm just kidding. Bottom line is I wanted to show you just some of the new work as a lead into Allison's really great single-cell work, some of the work that we've been doing with enteropathies, and give her a little bit of extra time as well. So here's what we did, again, sticking with my theme of it has to be published. Some of you probably read or saw us present this work in Philadelphia. Those of us that were there were thankful to survive another trip to Philadelphia. But while we were there was some good science. And in this particular one, we chose to use transcriptome of PBMCs just because it was now affordable. And so we're basically sequencing all cat body parts right now at Dr. Dow's lab and our lab. But I thought, well, let's go ahead and do the healthy kitties. So basically, we're using the bulk RNA sequencing just to see if we can move the needle with things like probiotics. This one happened to be an SF68 with psyllium study. That's a product in some of your countries. So basically adding a probiotic to that bacterium. Now it's a symbiotic. So this is a really big study that I look forward to you seeing all of it, but I wanted to not jeopardize publication. I'm just going to show you the parts that we've shown as an abstract so far. So basically what we did is we basically have a control group of kitties that got everything that's in that product other than the bacterium. And then the other group got the product. And they don't come to us in sachets. They come to us in capsules with either a red dot or no dot. And then we try to keep everybody masked to what is even in the products. And I often don't know until the end either. And then we, of course, culture to make sure that we have the predicted number of organisms mid-study, end of the study, et cetera. And so with this bulk RNA sequencing paper, it wasn't a ton of things, but there were definitely, between the groups, significant differences. When we then looked at the millipore, the chemokine-cytokine panel that many of us have used, I think Ebenezer helped with the development of that panel, we actually saw two things that were a little bit interesting. The TNF-alpha kitties, which are, as you guys know, cats that are healthy on the same diet living in this environment, they're not very immunologically active. And so a lot of the analytes you can't detect at all. But then the ones you can, like in this case, TNF-alpha. But why that was important is from the bulk RNA sequencing, the pathway analyses, we also showed this favors TNF-alpha pathways as well as TNF as well. So it has antiviral activities, which then perhaps partially explain why we saw the effects that we did in the herpes virus model, perhaps if you equate the cat to a small dog, then in the dog studies as well. So this part, I don't think anybody's ever seen yet. I did choose to put this slide in just because I thought it was a nice closing slide to see if there are a few questions before we bring Allison up. That feline macrophage cell line, it's a little tricky, it's a little temperamental to grow macrophages of cats, and not all feline infectious peritonitis viruses will actually infect cells in culture, which is often a problem for us, right, to study these new field isolates as FIPV. But we do have one that will grow well in cell culture. So just wanted to show you, these are just two ways of measuring cell death. So if you look at the control bars on both charts, those are cells grown without anything, and so 100% cell viability. If you go then to the next bars next to the controls, this guy right here, I don't think I turned on the pointer. Here we go, hang on just one second. Thought I was going to turn on the pointer, I did not. Try this one, there we go. Yeah, so if you look right next door, those are the cells that were just infected with a cell tropism FIPV virus, so you kill a lot of the cells. And then we had the serum, by the way, this was serum samples from the cats that had been supplemented for 28 days. And so then the serum, then different dilutions were evaluated, but we looked at the ability to block FIPV kill by the serum of those kitties. And the symbiotic group, group B, had more viable cells. And this work is going on still. This is just what I showed, just finished a while ago. We're doing larger numbers of the cats. This is just, I think, 4 of the cats in each group to generate these charts. P-values didn't make statistical significance. We're finishing that work now, and the new work is actually with herpes virus. Since we've shown some effects with that probiotic for herpes, we're doing a similar, just because herpes is a lot easier to grow on cells. And so hopefully if I ever get to come back to Can Summit, I'll share some of that in the future. I don't know what you guys were thinking after seeing that, is if that turns out to be statistically true, then I was quite intrigued because GS drugs are expensive. We're getting GS drug resistance. The holy grail still with FIP, for those of you that are not veterinarians, is if we could keep them from mutating. Probably never get rid of coronaviruses, but if we could lessen their mutation potential, that would be a nice thing. And so just to let you know what we're doing this summer is we're doing pedigree cats, quantitative PCR shedding levels with and without the probiotic, with the thought being is if you can lessen replication of enterics, you'll lessen mutation, right? Because you have to replicate to mutate. So that'll be a fun one as well. Got to have a really gung-ho vet student to do that one this summer. So look forward to sharing some new things next year. But to kind of close out then, for me, it's just been kind of fun now to try to come up with some clinical trials that a systemic immune stimulating something would be interesting to study. I've thought of many new ideas already just from being with you guys for the last day and a half, and I can't wait to get home to start writing those things. Well, I didn't necessarily want to go home, but going home and writing those up will be 2 different things then. But again, now we're getting a little bit more multi-genomic. We've got multi-omics. We're being able to do more things at the same time at an affordable price. And after a couple of questions, I really would like to invite Dr. Manchester to the stage.